So welcome to this short ReadStore tutorial where we want to upload a couple of FASTQ files into our ReadStore app. What we're going to do is first run through the whole process quickly, which will basically cover most of the cases in your everyday workflows. And then the second part of this tutorial, we're going to have a deep dive into the different options you have in uploading and staging FASTQ files. So let's get into it. We have a set of FASTQ files here from 6 datasets. These are paired end data for tumor and normal tissue, RNA-Seq, and what we want to do now is upload those files. So for this we move into our terminal and we navigate to the folder that contains the gzipped FASTQ files here. The format doesn't really matter, just needs to be FASTQ. And what we're going to do first is check if our ReadStore command line is available. For this, we just type ReadStore -H, and we see that we have the ReadStore client configured. And then all we need to do is to run ReadStore upload and define the file we want to upload. We start with uploading this replicate1 file. We confirm this, and then the client will perform the upload of the file. And once this is done, we get a confirmation. We can also make this a bit faster. We have a lot of files here, so we can just also say ReadStore upload and then expand with a wildcard here, and it will start to upload all the files. In the meanwhile, we logged into our ReadStore app and moved to the staging page. This is where we're going to find all the FASTQ files that we just uploaded. What happened in the background is a QC check for each FASTQ file, so the software makes sure that it's really FASTQ format and calculates some statistics like the number of reads or the read length or the quality scores. More to this later. And what we need to do now is we need to check in the FASTQ files in the staging area. And this check-in procedure is meant for grouping individual FASTQ files into datasets. The reason why we need this is that sometimes you have paired end reads, for instance. So you have basically 2 files that would actually make up your dataset. Sometimes you have just one. You may also have index reads, depending on your application. And what the software is trying to do is automatically group these FASTQ files based on the file names into datasets. So here, for instance, we see that this seems to be okay. So we have these replicate1, read1, and read2 files, and they make up this dataset, tissue normal replicate1. This is actually okay for most cases. If there are problems with these groupings, again, we will go through this later. But for the moment, what we can do is simply check in the file. This will open a dialog where we can assign a project, for instance. We prepared here this cancer project. This helps to group datasets later. Then there are other things that we can set. We will have a look at this also later. And then we can just hit the confirm button, and this will perform the staging operation for us. And now we see that it's only 10 datasets left. We can do the same thing with the next dataset here. We press the check-in button, but now let's say we want to change the name here, and we want to call it tissue healthy. Then we could do this here in this dialog here. Again, we are going to assign this to our cancer project and confirm the check-in. And that is basically all there is. These datasets now appear in our dataset overview, and that was basically all there is to upload and check in the datasets. And we are now going to look into the details and a deep dive into the staging procedure. So this overview summarizes the steps that we just took. What we've seen is that we started in our command line interface and ran this ReadStore upload operation. This operation is uploading and registering the files in a staging area of our database. And what is happening in the background after you call this command is the QC step. For those people that work a lot with NGS data, there's a software that's called FASTQ that many people use, for instance, that is calculating the statistics on the FASTQ files. And this QC step is doing something very similar. So it's checking that it's really a FASTQ file that you upload and that it's not corrupted. It's collecting the FASTQ quality, the thread scores, and then collecting statistics such as the number of reads. Finally, it's also performing a compression if you don't provide files that are already compressed. So plain FASTQ files will be compressed here because you want to keep the file size down when you store them later. On the staging area, and this is where the files end up in our user interface in the staging page, we have the opportunity to group FASTQ files into datasets. As I said, the reason for this is that we want to work with a dataset and not with an individual read1 or read2 file. This just makes things easier, and in practice you would hardly ever analyze just a read2 if you have paired end data, for instance. So this is why we group the reads depending on what type of platform and assay we use. Mostly, I think we have paired end data, so we group the read1 and read2 into a datasets and then perform this check-in in the staging. And this is actually meant as a QC step so that you are not uploading accidentally files into your database that are actually not of proper quality or not what you really want. This is why we have the staging area. And if you check in your files, they are basically uploaded or registered in the database and available then through the command line interface, through the SDK for users in projects and so on. We are back in our user interface, and what I want to do now is go through some of the details of the staging process and some of the options that we have here to inspect our datasets and to rename them, for instance, and regroup them. So again, this is where we ended up before. We are here with 8 datasets left, and first we're going to have a look at all the tabs we have when we check in this dataset. So we have now this tissue normal replicate 3, and as we've seen before, what we can do here in this first project tab is assign them to a project. This helps to keep a grouping of the datasets that somehow belong together. And then we can have a look at the other tabs. So we have a feature section here that gives us the option to add a description to this dataset. So we can maybe say that this is a normal tissue. Then we have a metadata section, and metadata is key value pairs that are stored in the database, and that should help you particularly to guide your analysis. So these are typically categorical variables that you can later use to reference these data. So examples are, for instance, the replicate here or the tissue. In this case, these metadata are inherited from the project. So you can set metadata keys in the project, and then all the datasets that you assign have these pre-filled here so that you can build yourself some sort of structure. So let's say this was our replicate number 3, and the tissue here was normal. And for this one, we can also add another metadata. So let's say this platform here was RNA-Seq, and so on. And like this, we can build these key value pairs that we can later on, if we do an analysis, easily access through the CLI or through the SDK, and those can then guide into the analysis, and we don't need to manually read them in anymore. This is very helpful. And we can also select some attachments here. So here we have a drag and drop area where we can upload files. So maybe we have some experiment details that we want to store, or some QC reports from our library preparation, or a sample sheet, anything like this. Any type of file can be stored here in the attachment, and we can also later access them. And then we have the read1 and the read2, and this basically gives us access to the statistics that are related to the FASTQ file. So here we have the output from the QC step, for instance, the number, the read length, the number of reads, the size, a checksum, and on the other side we see the thread scores plotted, so the quality across the different base positions, and as usually it's dropping a little bit in the end, and the same also for the second read. And inspecting these is of course important if we want to make sure that the quality of the datasets that we upload is consistent. And when we are fine with this, we can again press the confirm button and it will again stage this dataset. So let's discuss some cases that can happen that are related to the naming of the datasets. Because as I said, what the software is trying to do, it's trying to automatically infer the dataset name based on the name of the FASTQ files. But sometimes the names of the FASTQ files that are uploaded are not really descriptive. So if you name your FASTQ files test or whatever consistently, then this may not help you to build dataset names that are meaningful. And I prepared this example here where we see that we have now 4 times tumor replicate 1 and each 2 pairs of read1 and read2 files. And this can, for instance, happen if you upload these tumor replicates from different folders. And then the software, because it looks at the name of the FASTQ files and not at the file path that it uploaded, may not be able to distinguish this. So of course, in our dataset collection, the datasets must be named uniquely. And so for this, we now need to perform a name change. We can very easily distinguish which FASTQ file came from which source, because we record the upload path here. And here in this overview, we can already change the naming. So let's say the first pair is basically from our patient A. Then we are just going to change the dataset name in the table here directly, confirm this, and what will happen is that the software is going to create a new entry here, only with the single FASTQ file here based on the new dataset name. We now copy this and take the associated read1 dataset here and also change this name. And then the software is going to build us a new dataset based on the changed dataset name. With this, we then have a correct pairing and we can check this in, and this will also make our dataset name unique so that it will be accepted by the database. We can also change here the names of the FASTQ files. It's not so super important because ideally you're not dealing directly with the FASTQ file names anymore later. You actually don't have to. We can also change the read here. Sometimes the database is not able to find the automatically detect what read it is. If it's not somehow a part of the file name. And then you can assign this here in this dropdown. You would see an NA here. And like this, you can build your own dataset naming schemes. And finally, let's check how we can get rid of datasets that we accidentally uploaded or that did not pass QC or that we want to get rid of for any other reason. For this, we can simply press this delete icon here and then click the confirm delete. And this will basically remove the datasets from our staging overview. There's also a search functionality here. So if you have lots of datasets in your staging view, you can search for dataset names using this search bar here. And I think those were the main functions of the upload and staging process. If you have questions or comments, please leave them under the video or reach out to me. If you want to use ReadStore, you find more information in the video notes, and I wish you a great day.